Hello everyone, let's talk about DNA replication today. So as we all know that DNA, this is your DNA, it's a double stranded structure and it has got two ends, they are the 5' end and the 3' end. Okay, and there are numerous DNA bases which are present which are paired like in C would be paired to G, that is your cytosine would be paired to guanine, and adenine would be paired to thymine. So that is your CG and AT ends over here which are present. Okay, so this is your basic double-stranded DNA structure. So now we are talking about how the replication works. So the first step in this DNA replication is to separate the two strands. Now how these two strands are separated, that is by your enzyme helicase over here which is present. Okay. Now this unzipping is done and the DNA that was double stranded is now divided into two parts, that is your two single strands, and this fork which is present over here which is formed after the function of enzyme helicase is called as your replication fork. Now, each of these separated strands provides a template for creating a new strand of DNA. Now, this is running towards this part from the 5' end towards the 3' end, and the first function is by the enzyme primase over here. Okay, now this enzyme primase starts with the process. What happens over here is this primase makes a small piece of RNA which is called as a primer. So this is your primer over here which is a starting point of the construction of new structure of. Now once the primer is formed, an enzyme called DNA polymerase that binds to the primer and will make a new strand of DNA. Now, one point that you need to remember is that this DNA polymerase over here can make the strands from that can add the DNA bases in one direction only, that is from 5' end to the 3' end over here. Alright, and due to this, there is a strand which is formed continuously over here, and that's why it is called as the leading strand. So, this leading strand is formed continuously without any interruption, and this is your by the action of DNA polymerase. Whereas when we talk about the other end, this part, this chain, that is your lagging strand. Why this is called as lagging strand? We will talk about because it is not formed continuously as it runs in a opposite direction. So due to this, the DNA polymerase enzyme over here can make this in the strands in small chunks only. So these are the small chunks which are made, and these small chunks over here, they are called as the Okazaki fragments. Now each of these fragment is started with a RNA primer, and DNA polymerase then adds short row of DNA bases in 5 to the 3 direction, that is your opposite direction. This is how this goes in the opposite direction. Then further the next primer is added down the lagging strand and another Okazaki fragment is then made, and the process goes on repeating over here. Now, once the new DNA is formed, the enzyme exonuclease removes all the RNA primers from both the strands of DNA. So this is your exonuclease enzyme which removes the strands of DNA, sorry, the primers from the DNA, and then another DNA polymerase is released which fills the gap which are left behind over here, and hence a continuous band is then formed. So once the complete band is formed, then comes the role of these two enzymes over here. These are your DNA ligase. Now what is the role of DNA ligase? This seals up the fragment from both the sides, and hence from one DNA, one double stranded DNA, we can get two DNA, and this is how the process of DNA replication works. Now you should remember that this DNA replication process is a semi-conservative procedure. Why semi-conservative? Because from each of the double-stranded DNA what we get two new DNA, it has got one part from the older chain and one that is totally newly made. Okay, thus it is a semi-conservative procedure or process. So this is how DNA replication works. Thank you.